Before we can start talking about genetics, we need to talk a little bit about meiosis because genetics is the study of inheritance and we inherit our traits through our gametes from our parents. During the process of meiosis, gametes are going to form and there are 4 possible outcomes, any one of which could be the gamete that is used to produce an individual. In each of our cells, we contain an entire complement of the genomes from each of our parents. So what this means is we get 1 copy of each type of chromosome from our parents. One set of copies comes in the sperm, 1 set of copies comes in the egg. In this image here, we have a cell that has 2 copies of 1 type of chromosome. And we see that on this chromosome, it has the G gene and 2 different copies, a big G and a little g. We'll talk about those in just a little bit. In preparation for meiosis, those chromosomes replicate and then they are separated in 1 round of division. In your semester, if we haven't done meiosis yet, don't worry too much about this process. We're just trying to get to the end result. During the first round of cell division, we separate homologous chromosomes. Now this means we separate these 2 copies, one that originally came from each parent. In the second round of meiosis, we separate our replicated copies. And so what we end up with is 1 copy of each type of chromosome in each of our gametes. So the options here are that the gamete has acquired the big G allele or the little g allele. And 50% of our gametes are going to have one of the parental alleles, the big G. 50% of cells are going to have the other parental allele. Now, any one of these could go on to be the one that is used in a fertilization event. Excuse me. And if we think about this happening for the sperm and the eggs, if we start with the same genotype for each parent, then each parent could potentially produce a big G or a little g. Now, what happens in the parent, what they're able to pass on, depends on their particular genotype. But right now, we're just going to go with they're each heterozygous big G, little g. So during the process of fertilization, that sperm that has been produced from meiosis and could have either a big G or a little g, depending on which one gets passed on, and that egg, which could have a big G or a little g, depending on what gets passed on, are going to come together. So our options here for gametes are that this sperm has a big G or a little g, and that this egg has a big G or a little g. Let's say just by chance that it is the big G in the sperm that's passed on and the little g in the egg. Now I say by chance because any one of the 4 gametes could be released, could be the one that's involved in this fertilization. When they come together, they produce a fertilized zygote. So what we actually have here is that original parental chromosome, 1 from each parent, and now the offspring has acquired that new combination of chromosomes. Just by chance, I happen to have picked that the individual becomes heterozygous. It can actually be any combination of big G and little g, depending on what comes in the gamete. So then after fertilization, the zygote divides, becomes an embryo, and eventually grows into a little human or a little plant, depending on what we are talking about. We're going to be talking about plants today, so I went ahead and wrote down plants. So in the zygote, we find 1 copy of all the chromosomes from the male and 1 copy of all the chromosomes from the female. All right, during fertilization, these gametes come together in any chance combination. So we started with big G in the sperm, big G in the male, and he produced two different gamete options, big G or little g. And then for the female also, she could have produced big G or little g. So the one that actually gets used is just going to be based on the flip of a coin. Whichever egg happens to be released, whichever sperm happens to be released. So we can determine what combinations are possible by something called a Punnett square. And we're going to come back to that in just a minute. First, we're going to talk about a couple of important terms and a couple of different patterns that we might see as a result of those Punnett squares. All right, two terms that we're going to see all the time are dominant and recessive. So the dominant allele is always going to be written as a capital letter. So in this case, we've got our big G, and we call the allele that covers up the other one the dominant allele. This is important for heterozygotes only because our options are going to be that they are homozygous for big G, heterozygous for big G, little g, or homozygous for little g. So in the heterozygote, the phenotype that we see is going to come from the dominant allele. So let's say that big G stands for green plants and little g stands for albino plants. If green is the dominant, this means that in the heterozygote individual, they are going to appear green. The recessive allele is the one that is covered up by the by the other allele in the heterozygote. So this means that in the heterozygote, that little g is not seen. We are going to call that little g, and we are going to always denote our recessive as a little g or a little allele. It really doesn't matter what letter you use for this. You could use A, you could use B. Generally, you pick something that is related to the trait that you're looking at. So an A or a G would be appropriate here. And also in general, you want to try to find something that looks different in in capital or lowercase. So for example, C is not a great choice. O is not a great choice. P is not a great choice, although our lab does choose P for the second exercise. All right, a couple other things because they're going to come up in the lab at some point. True breeding means individuals who are homozygous. So if we say true breeding parents, they were the homozygous parents. The F1 generation is the first generation of a cross. So if we cross individuals who are big G, little g, their offspring are going to be the first generation. This was originally in reference to true breeding parents in Mendel's experiments, but it's not necessarily limited to that now. It could be any parental cross and the first generation. The F2 generation is a second generational cross. This is done by crossing two F1 siblings. This is generally used in plants. It's not usually used in animals, but its purpose was to determine which trait was dominant and which was recessive. We're going to see 4 different inheritance patterns in the next week or two. The first is complete or simple dominance. So this is kind of your normal or standard. This is when one trait completely hides the other in the heterozygote individual. So this is where we're going to use our capital lowercase designation. So when you see that capital lowercase, you're going to assume that it is simple or complete dominance. Another possibility is something called codominance. This is where both of the phenotypes are seen in the heterozygote. So both of them are distinctly visible. So for example, we have this black and white chicken here. The checkered chicken is the result of a codominant allele situation. We use a slightly different notation here. We use 2 capital letters because they are both displaying their dominance. So in this case, we might say that the heterozygote is BW for black and white alleles, both being dominant. In codominance, we use different letters. In complete or simple dominance, we always use the same letter. Next, we have incomplete dominance. This is where neither allele can fully hide the other, but neither one is strong enough to fully be seen themselves. And so what we get is an intermediate trait. Again, we have a slightly different notation here. The notations for complete and incomplete dominance are, codominance and incomplete dominance are going to be a little bit different. There's no standard. The one that I generally use, just to show that there are differences in inheritance, are a superscript on a letter. So for example, the C might mean color. And so what we're saying is the purple color allele is one allele, the white color allele is the other. These plants are called 4 o'clocks, and they are a common example of incomplete dominance. So we have the purple flower here. We have the white flower here. So the homozygous situation for each of those is going to give purple or white. The incomplete is going to show up in the heterozygote, where we have this sort of intermediate trait where we get a light purple that is sort of a mixture between purple and white. Next week, we will also add in sex linkage. We're going to look at two types of crosses today. The first is a monohybrid cross. This is where we are going to look at the interaction of one set of chromosomes, and we're crossing one set of alleles for one gene. So we are going to be looking at the green and albino plant allele. This is an allele that helps produce the chlorophyll pigment. The white ones are white because they have no chlorophyll, which also means that they aren't going to survive very long because they have no chlorophyll. They can't perform photosynthesis, so they can't get sugar from anywhere. So the big G allele is the dominant allele. The little g allele is the albino allele, and that is recessive. So these individuals up here are the offspring. And the first question in your lab is going to ask you, what is the likely genotype of each parent? So you have to think about what genotypes from the parents would give offspring that are both green and white. So you have to think about our definitions for dominance and recessive alleles. And you have to think about what parental genotypes would ultimately result in producing both green and white offspring. The question asks you to write out all possible genotypes crosses for the two alleles. So for example, one possible cross for the parents could be big G cross little g, little g. So you have to think about all of the potential genotypes for the parents, and then you have to pick out the most likely one. So sometimes students will immediately identify which one this has to be, especially if you've had biology with genetics before. What we're asking for in that question is, doesn't matter what you think the parents are, what are all of the potential possibilities? And then you circle the one that you think these parents were. When we perform a monohybrid cross, the first thing we need to do is figure out the parental genotypes. Sometimes this will be given to you, sometimes you'll have to figure them out. The next thing we're going to do is make our gametes by segregating our alleles. So for our first parent, our gametes are going to be big G and little g. Those are the two potentials that that individual could make. And then our other parent, big G, little g. Those are the two potentials that that parent can make. Next, we're going to fill out our Punnett square. So this is a 2 by 2 square. And it's just a way to determine all of the possible combinations that we could get from our parents. So we're going to take the first set of gametes, big G and little g, and we're going to place them on our Punnett square. Then we're going to take our second set of gametes, big G and little g. It doesn't really matter which parent is which. You just want to keep the gametes for each parent together. Next, we're going to make all of our possible combinations. So we're going to do this by combining the top and the bottom, kind of like a multiplication table. So if we combine the big G gamete with the big G gamete, we are going to get a green individual. If we combine the big G with the little g, we get a heterozygote. And because this is complete dominant, we're going to see that big G allele shine through. So this is a green individual. And then if we do this big allele with this little allele, we get another green. And then lastly, if we combine the little g allele with the little g, we get an individual who is white. For every cross that we're going to do, we're going to write out the genotype and the phenotype ratios. So these are the genotypes inside your box, and these are the phenotypes, the physical characteristic that you're going to see. So our genotype ratio for these individuals are going to be, for every one homozygous dominant, we have 2 heterozygous individuals and 1 homozygous recessive. We're going to do the same thing for the phenotype ratio. For every 3 green individuals, we're going to have 1 white individual. Now, this Punnett square also tells us probabilities. So there is a 1 in 4 chance, or a 25% chance, of having a white plant. So if our plants were to have exactly 4 offspring, we could expect there's going to be 75% green, 25% white, or 3 green for every white plant that we produce. But this can be extrapolated for a higher population number, because it's very unlikely that a corn plant is going to have just 4 individuals, or it's very unlikely that a family is going to have exactly 4 children. So we can use these probabilities for a higher or lower number of offspring. So if we were to have 100 corn kernels, we would expect that 75 of them, or 3/4 or 75%, are going to be green, and 25 of them, or 25%, 1/4, are going to be white. And we can determine that for any group of offspring that we have. We can also look at a dihybrid cross. This is a cross for 2 genes on 2 different chromosomes. So we're going to be looking at corn color again. Corn color, but we're going to be looking at the kernel colors this time. And then we are also going to be looking at the shape of the corn kernels. So our color options are purple or yellow, and our shape options are smooth or rough. So smooth is kind of your normal corn shape. Rough is kind of a raisin-shaped corn. It's not the same thing as drying out a corn, but that's kind of the shape that we're going to get. So we need to figure out all the combos of these 4 chromosomes that we could get in our gametes, keeping in mind that each gamete is going to get 1 copy of the P chromosome and 1 copy of that R chromosome. And that each gamete is going to get 1 P allele, either the big P or the little p, and 1 of the R alleles, either the big R or the little r. So we're going to borrow a process from math. We're going to borrow the FOIL method from math, and we're going to start with our parental genotypes. So for this cross, they are going to be big P, big R, or the heterozygotes. And we are going to use our FOIL method. So foiling is for multiplying parentheses. We're going to use the same idea here. So the F stands for first. So we're going to take our first 2 alleles. That's 1 gamete. Outer, our outer 2 alleles. That's another gamete. Inner and last. Those are our 4 alleles. So I'd like you to try this for the other parent as well. You can do this for any parental genotype combination. Just keep in mind that they're going to get one of the P alleles and one of the R alleles. You should not have any more or less than one each. So let's go ahead and look at what this looks like on a Punnett square. You saw in the previous slide that we got 4 gametes from this parent and 4 gametes from this parent. Again, we used our FOIL method. First, big P, big R, outer, big P, little r, inner, little p, little r, and last, little p, little r. So we can do that for our other parent. They're going to give us the same gametes. And now, just like our small Punnett square, we're going to combine our alleles. When we do this, we are going to keep our P's together and we're going to keep our R's together. And for each one, we're going to determine the phenotype. So this one is going to be purple and smooth. Let's go ahead and do another one. I'm not going to do all of them, but I'm going to give you an example of each one. This one is going to be purple and rough. This one is going to be yellow and smooth. This one is going to be yellow and wrinkled. So I would like you to try this. Fill out all of your combinations, fill out all of your different phenotypes, and then you're going to write out the phenotype ratio. You don't have to do the genotypes for this one because it gets messy, although it would be great practice. Once you do this, you're going to tally all of your different phenotypes, and you should get a ratio that is 9 to 3 to 3 to 1. But I want you to label, so 9 corresponds to which phenotype, 3 which phenotype. And we never want to leave our labels naked. We always want to, our ratios naked. We always want to leave a label on them. In the next slide, I'm going to show you the completed Punnett square. I want you to check that yours is correct. Notice that I have tallied up the phenotypes and I've written out what number corresponds with what phenotype.